today we are going to be having our second webinar in our series for the Industrial Biotechnology Innovation Catalyst. So for those that don't know, or maybe I can just remind you briefly. So the Industrial Biotechnology Innovation Catalyst is an EPSRC funded impact acceleration account grant. It is there to help support the translation and exchange of knowledge from our higher education sector across the northwest of England out into industrial partners, whether they be large corporates or they be micro companies that have just started and are just starting to grow. That's where 2/3 of our 5 million funding are being channeled into, and we've got some grant calls open at the moment. So if you want to check out our website, I'm sure Jen, over the course of the webinar, Jennifer will be able to pop our funding web link into the chat. So please click on that and have a look to see what we've got available at the moment. So that's 2/3 of the money. 1/3 of the money is there to help support and catalyse the impact creation from research from our higher education institutes into new startup companies, into spin-outs, and to help support new initiatives coming through along the way. And we've got activities on board, and for that we've got our pitch day for our currently dedicated iCure programme on Friday, actually, which is great because it's the entrepreneurship week this week. So excellent timing on behalf of the IBIC team. So today, really excited and really pleased that we've got two speakers today. We have Neil Dixon, who is an academic within our Manchester Institute of Biotechnology here in the University of Manchester. And we have Michaela Chacon, who is one of Neil's postdocs and is actually a poster award winner, I believe, from one of our last conferences. So we've got an award-winning team to talk to you today, from waste to wealth. And they're going to tell you a little bit about their research and what they're up to help create sustainable impact from their research work. So Neil, I'm going to stop talking and hand over to you to take it from there. So thank you very much, Neil.
Okay. Thank you. How's that? Are we good? Awesome. Okay, thanks very much for that very warm introduction to us all. Ellie, hope you do you justice. So yeah, we are based in the Manchester Institute of Biotechnology. We're interested in engineering microbes to do useful things. A large section of that is about how we can try to close the loop and use biotechnology to both make chemicals and biopolymers from renewable sources, and also engineer microbes to allow us to deal with these things at the end of their life, try and create new biopolymers, new materials with better end-of-life properties. So I'm going to start off with this slide. It's commonly used, but I quite like it, and it frames a lot of our current thinking and the way that we try to approach these things. As we know from this data that comes from the Nova Institute in Italy, that the demand for embedded carbon is going to double in the next 25 years. So this is carbon that goes into materials and products. It's not carbon that goes into bioenergy applications. So it's the materials, the carbon that goes into our plastics, into our chemicals, into our polymers, into our clothes, into our medicines, into our insulate our homes and so forth. So it's really important for the lifestyle that we lead. So the prediction is that we're going to go from a position where we are currently, where the carbon almost exclusively comes from petrochemical sources. And then within 25 years, we're going to massively pivot and come from this whole array of different carbon sources, some from bio, some from off gas, some from recycling. However, this is a really complex change and dramatic shift. So not only is this going to be new feedstocks, it's got to be feedstocks that are not pure. They're going to be seasonally variable. They're going to have co-contaminants in there. They're going to have high moisture content, and it needs new infrastructures, new workforces, and new technologies to allow these things to happen. So obviously, being a biotechnology group, we believe in the power that biotechnology can play in a range of these sectors. So the brief overview of what I'm going to talk to you about today over the next sort of 15, 20 minutes or so is some of the technologies and some of the genetic tools that we've been using and implementing that allow us to engineer microbes So firstly, around how we've applied these approaches to engineer microbial systems for valorization of waste biomass, biogenic feedstocks, how we can convert that into value-added products, and then moving away from sort of natural polymers into sort of xenobiotic type polymers, so polymers that are made by humans, partly on plastic, and then very briefly I'll just touch onto the concept of using alternative types of plastic, alternative types of polymers that are from human waste, and that will set up Michaela's follow-up talk that will follow mine. And at the end, I'm just going to sort of try to contextualize this in terms of some of the challenges ahead and some sort of stakeholder engagement activities we've been doing through some Manchester funding and partly funding more recently through IBIC in terms of engaging with regional stakeholders in the chemical manufacturing sector. Okay. So let's start at the beginning. So we've been working with researchers out in Brazil for the last sort of 15 years or so. So arguably Brazil is the leading example of industrial biotechnology that interfaces with consumers directly. So Brazil produces bioethanol that is used to power the domestic vehicle fleet. It does this by using the sugar juice from sugarcane. This feeds industrial scale fermentation, where yeast is used to produce bioethanol, which is sold at very high percentages bioethanol content at the petrol pump directly to the consumer. So up to around 90% of fuel at those petrol pumps is composed of bioethanol. And although they're not the biggest global producer of bioethanol, I think the fact that actually 90% of all domestic vehicles in Brazil are converted to be able to run on this flexible fuel approach, so very high concentrations of bioethanol, is why they're the leading example of industrial biotechnology adoption. However, there is still waste from this process. They have these big mountains of residual biomass, these lignin bagasse mountains, very rich in residual sugars and aromatics. So we have a project some time ago now how we're trying to apply industrial biotechnology, engineering, biology approaches to valorize some of this waste material and convert it into higher value materials such as flavors, fragrances, and active pharmaceutical ingredients. So we're really interested in microbial genetic regulation and how we can use these native sensing mechanisms and hijack them for our own purposes in biotechnology. So nature naturally uses sensing mechanisms to turn on different processes, either to swim towards nutrients or turn on particularly biological cascades. And it does this by sensing certain molecules. In this context, sensing aromatics and sugars, which can come from biomass. By developing genetic circuits, we can turn on output reporter genes. So for example, a gene that codes for a fluorescent protein. This will allow us to monitor that biocatalytic process, screen for new enzymatic function, either new enzymes or evolving enzymes through functional metagenomics or directed evolution. And these concepts also allow us to evolve strains or to maximize or optimize flux through biosynthetic cascades. So one such system we developed, just to give you an idea of the nuts and bolts involved, is one of these systems here. It's a transcription factor based system. This particular biosensor responds to this dihydroxybenzoic acid, and it's a simple repression derepression system. So in the presence of the effector compound, the system is turned on and you get green cells, green fluorescent protein, which you can see in this dose response curve down here at the bottom, and we can do a series of structure activity relationships. So we're going to come back to that briefly. So that's some of the ideas and the approaches that we do in terms of genetic regulation. Shifting now in terms of some work that we've been doing in terms of biological catalysis and the ability to use this approach to convert low value agricultural waste into high value compounds. So here we have a schematic on the left hand side of lignocellulose, which is a hemicellulose fraction of biomass. So xylose sugars linked to ribinose, ester link through to this frulate compound here. So if we hydrolyze this ester bond and then reduce it down, we make this coniferol compound. It's a high value compound itself. It's also an intermediate into a range of flavor and fragrance compounds and active pharmaceutical ingredients. So some time ago now, we developed a biosynthetic cascade for this that involves two different enzymes that reduce this acid down to the alcohol CAR and an AKR. And in monoculture, in microbial cultures, we got around a gram per litre of these products directly from biomass-derived aromatics. So non-optimized system, reasonable initial titers. We then took that concept further forward. Obviously, this is a two-stage process where you need to release the aromatics from the biomass and then carry out an enzymatic or microbial biotransformation. We want to see if we can consolidate that all into one process. So we engineered two microbial specialists. One we're equipped with hydrolytic enzymes that would be expressed and secreted into the extracellular space, degrade down the biomass, release nutrients and aromatics. And then in a second microbial specialist that would transform those compounds into value-added materials. And we've done a series of that, which you can read down this paper down here. Just to show you the concept, what we've done, we have in an aqueous phase, the biomass, the two microbial engineered specialists, and then an organic top phase that allow us to facilitate that downstream purification. So we can extract the more non-polar products, in this case, cavicol, which is one of the compounds we're making, and that extracts into the organic phase. That drives the equilibria, but it also, as I say, facilitates downstream purification. Okay, so those are just a quick snapshot of some of the work that we've been doing in this space. I'm going to quickly shift on to non-human derived, should we say, or xenobiotic derived polymers, and how we can potentially use the carbon in those materials for microbial growth and bio-based production. So as we know, plastics is a really essential material for everyday life, for these very different applications we've been talking about. Great barrier properties, really reduces some of the greenhouse gas emissions associated with transport because it's lightweight. However, we're really not very good at dealing with that plastic at its end of life. And we've really made so much plastic now in the last 70 years, it's really quite astounding. So this data that comes from this study from Ron Milo's group reports that we've made so much plastic now that the mass of that plastic is double of all the animal kingdom. So that's all humans, all wildlife, and all livestock, which is really quite an astounding piece of statistics. So we need to think about our end-of-life solutions for plastic. So one of the ways you can potentially think about using the carbon in that is by using microbial metabolism. And here's a schematic of one of the ways that polyethylene terephthalate, so this is the plastic that's in our plastic bottles, but also in our clothes, commonly called polyester, can be degraded down into its constituent components, here TPA or terephthalate and ethylene glycol, and transported inside bacterial cells and allowed that to be broken down into central metabolism. So a few years ago now, we were interested in the transport process of this TPHC protein. So we did some structural biology where we co-crystallized the transporter in the presence, the absence of the terephthalate compound. So you can see that in the yellow here in the active site, this ligand bound and closed that allows that substrate to be delivered to the membrane and transported across. We're also interested in this kind of catabolic operon, the arrangement of it and its genetic regulation. So we did some bioinformatic mining and pulled out a series of homologous operons from the databases and pulled out some of these regulators, these TPHRs, and constructed sort of biosensors. So these are systems that respond to terephthalate here and would turn on an output gene, a green fluorescent protein. So you can see that from that primary screen, how we pulled out a series of these here in this plot. We then optimize these systems by developing a series of different biosensors. We modularized them. We incorporated different strength ribosome binding sites and promoters. And essentially, this allowed us to change the stoichiometry of these different genetic components within this genetic circuit. And here we employed an approach called design of experiments that allows us to very efficiently sample that vast multifactorial space. So we had a series of these RBS promoter libraries. If we'd screened and generated all of those, it would have taken us forever, but it also would have meant that we would have needed to make 1 million constructs. So using this design of experiments approach, we massively compressed down the experimental design that is needed, the number of designs down to 17 discrete designs. And also we could learn and interpolate from this. It's a model, we can understand how these systems are functioning. And we also came up with a couple of different biosensors that respond in different ways. One that's a very digital system, Small changes in effector concentration on this x-axis give a very steep or rapid response on that y-axis, and one which is more granular. And you can think of a number of different applications where these two different performance attributes will be required. So we use the digital system in a primary screening where we screened a series of lipases and esterases for their ability to degrade polyethylene terephthalate into its constitutive monomers, where we use the biosensor to read out the hydrolysis end product, the TPA release from that, and that identified this LCC cutinase as the most active. We could then take that forward and use a biosensor which has a much more analog or graded response and optimize enzymatic or environmental conditions. Here we can tune very closely temperature and pH to find out the optima in this heat map here, which is also validated by HPLC. So the last bit of data I just want to show you is where we've taken this further forward now, and we're thinking about using this TPA as a carbon source. So this is the catabolic operon that we've developed that allows this terephthalate to be degraded down into this intermediate, this dihydroxybenzoic acid or coteketuate. And that whole catabolic operon is under the control of this transcription factor that I told you right at the beginning. So it's a kind of feed forward loop. We need a little bit of flux through the pathway to turn on the system. And this is how that carbon from ethylene glycol and terephthalate can enter central metabolism. And so we incorporated these gene clusters, these non-native gene clusters, into our engineered microbes. So this is a microbe that can't natively grow on these carbon sources. And now we can show that for the first time they can grow on these plastic-derived carbon sources. So you can see the consumption of these two carbon sources down here in this sort of mustard and orange line, and the concatenant increase in biomass as that carbon is consumed and converted into biomass and energy for that microbe to grow. Then we wanted to see what was the highest value compound we could make. So we have in our freezer a series of previous projects, a series of genes that encode for human therapeutic proteins. So somewhat gimmickly, but really trying to push the envelope in terms of demonstrating how you can really convert waste end-of-life materials into very high value things. We installed the genes that encode for a series of human therapeutic proteins inside this engineered organism, the Pseudomonas putida, and it converted that carbon into biomass, into energy, but also building blocks to allow us to produce these therapeutic proteins. So essentially we can make treatment for type 2 diabetes. You can produce insulins, for example, directly from the carbon from waste plastic. So that's the last technical slide I want to show you about. Just very briefly, we want to talk to you about this challenge. We know we've got this linear economy. We need to move away from that for various different reasons, for various different climate change reasons, and think about using resources to source that carbon. We know that there is historical examples such as first generation biorefinery and energy crops that can satisfy some demands. Around the world, we produce 28 billion gallons of bioethanol from this route. However, if you look about the amount of land that's required for that and the amount of land that would be required to produce only even 20% of that future demand for embedded carbon, there's really just not enough arable land around. So we would need to use around 10% of all arable land just to satisfy our demand for embedded carbon, or just that 20% of the embedded carbon, that 200 megatons we talked about right at the beginning. So it's just not really feasible. So we need to think about where we produce carbon, where carbon waste is, and how we can process this, and then think about this in a much more cyclical manner. So I'm going to pause at that point on that particular technical topic, and then that will segue quite nicely, hopefully, into Michaela's talk. So just very briefly to wrap up, I kind of appreciate the time is ticking now in my allocated time. I want to talk to you about some of these more holistic or more systemic challenges. So we know we've got this technology, industrial biotechnology, biomanufacturing, whatever you want to call it, and its ability to utilize renewable feedstocks and produce the chemicals and materials we need in our everyday life. So we want to think, what are the challenges that are impeding that technology adoption? Is it technology readiness? Is it scale? Is it workforce? Is it infrastructure? Is it locked in commitment to other feedstocks and supply chains and so forth? So we've been trying to look at this in the round, particularly focused on the Northwest chemical manufacturing. So in the UK, the northwest of England is one of the 3 major chemical manufacturing sectors. So we've been doing some mapping, understanding the size of that, but also trying to link in the nascent biotechnological sector in this space, as well as thinking about other aligned sectors. So, for example, those producers and processors of waste carbon, and also sectors which we class as sort of traditional biomanufacturing sectors. So those in brewery, in food, and anaerobic digestion, where there might be some cross fertilization in terms of skills and workforce and supply chains. So this has been supported by University of Manchester Research Institute, UMRI, and also more recently directly from the IBIC. And the person who's been really instrumental in delivering all this has been a graduate intern, Ling Ling Bu, who's developed really and driven all this all the way through. So big thanks to her. So the one other project just before I wrap, I just want to highlight that's just been awarded is very much aligned to what we've been talking about utilizing waste carbon at the end of its life and using those as a feedstock. So plastic, biogenic waste, carbon, and C1 gas as a feedstock for biomanufacturing to produce monomers, polymers, and so forth, and develop innovation through that space. So this is a project that's primarily initiated through the National Science Foundation in the US. In the UK, our component is funded by the BBSRC. You can see the names of the people who on the screen are involved in this in Manchester and elsewhere in the UK, and we're actively recruiting in this space a number of different roles. So if you're interested, please do reach out and get in touch. So just to wrap then, we're really interested in using bioprospecting to find transporters, regulators that we feel are useful for bioeconomy, biorefinery, and biomanufacturing. We have a really strong interest in the development of genetic tools, these so-called genetically encoded biosensors, with their applications in sensing and regulation. And I talked to you around a couple of different processes very briefly, both monoculture and then partially also co-culture processes, where we've been trying to consolidate those processes or to reduce the number of unit operations. Okay, with that, I will just then acknowledge all the people who've had a part in all this, who've delivered the research. You can see various different photos over the years on the screen, various names, funders and collaborators, and finish there and I think pass over to Michaela.
Okay, perfect. I'll just share. Okay. Can everyone see this slide?
Yep, perfect.
Just go full screen.
You just need to put it, full screen.
Perfect. Okay. So, yes, I'm Mika. I'm one of Neil's postdocs. And what I'm going to tell you about is some recent work that we've done investigating the potential valorization of complex waste through combined enzymatic hydrolysis and catabolic assimilation by Pseudomonas putida. And if this is a story at the end that you're still interested in, you'll be able to find the paper that's attached to the story in Trends of Biotechnology in the new year. So, like Neil introed, and like we are all, I think, quite painfully aware, the majority of global supply chains are dominated by linear and carbon positive economic models that are just unsustainably reliant on finite fossil fuel reserves. And these systems have big environmental consequences by driving climate change through greenhouse gas emission and contributing to resource depletion. And in the 90s, an increasing awareness of just how unsustainable this model is spurred interest in replacing fossil fuel feedstocks with bio-based ones. And this led to the emergence of the biorefinery concept. And the idea here was to create an analog to petroleum-based refineries to afford the production of fuels and chemicals and materials from renewable feedstocks in a more sustainable manner. And often you use microbial production workhorses for this work. In its first iteration, Biorefineries relied on biomass feedstocks that have a high starch content, like corn, sugar beet, and rice. Neil touched on this as well. But these feedstocks have a fatal flaw, and that is that they are also food. And this created a fuel versus food dilemma. And ultimately, this dilemma drove a necessary shift towards second-generation feedstocks. And what these are mostly organic process residues and wastes from agriculture, food industries, paper and pulp, etc. So things that don't compete with food and offer more material and carbon circularity. And what makes them particularly valuable as well is their abundance. So we generate billions of tons of these materials annually, which is currently wasted. But second-generation feedstocks do come with their own challenges. They are far more recalcitrant and compositionally complex and variable over time. And this poses a significant challenge to biorefineries because despite this high compositional variability, we still need to consistently and efficiently convert all of that input carbon to product for these processes to be economical. Biorefineries really need to operate at that intersection of precision and flexibility if we want them to be able to succeed. So this brings us to a core challenge, and that is, how do we design biorefinery platforms that are capable of handling this variability, ones that are flexibly able to accept a wide envelope of feedstock characteristics? And from a microbial bioproduction standpoint, this can be especially challenging because when faced with heterogeneous mix of carbon sources like that shown here, most microbes will consume them selectively due to incompatible metabolism or sequentially with long dioxic lag phases between preferred substrates. And ultimately, both of these behaviors can prolong cultivation time and reduce carbon conversion efficiency, which ultimately undermines the economics of a bioprocess. So to tackle this issue of selective utilization, many research groups have done some excellent work engineering bacteria to metabolize non-native waste-derived substrates. And these efforts have expanded the capabilities of microbial hosts by incorporating the catabolic pathways required for some lignin-derived aromatics or C5 sugars or alcohols, monomers from plastics, etc. However, consolidating all of these metabolic functions into a single platform host capable of robust and flexible and resilient metabolism is still in its early days, despite this being critical for process efficiency, cost, and scalability. And so that's what we were interested in investigating in this work. We wanted to explore the possibility of developing the bacterium Pseudomonas putida as a production host for the with the metabolic versatility and plasticity that is necessary to co-utilize variable and heterogeneous compositions of native and non-native waste-derived substrates, and also to consistently funnel that variable input carbon towards the formation of value-added products. So as a proof of concept, we engineered a strain of Putida to operate in the biorefinery pipeline that you see here. So using a complex feedstock input, we wanted to make 2 exemplar products, a low-value bulk chemical, specifically polyhydroxyalkanoates, and a high-value biotherapeutic protein called interferon. And these products were chosen deliberately. PHAs represent a renewable alternative to conventional plastics, while interferon demonstrates the system's ability to make high-value, specialized products. So the feedstock that we used for this work was a proxy for municipal solid waste, which is also referred to as MSW, and which I will probably refer to as MSW in this presentation. And what municipal waste is exactly what it sounds like. It's household and commercial and industrial waste that's collected by local municipalities. Currently, global MSW generation is estimated to be about 2 billion tons per year, and most of that goes to landfill, where it sits and it contributes heavily to greenhouse gas emissions, making it a vastly underutilized feedstock. So instead, by diverting it to biorefineries for valorization, we could potentially divert millions of tons of waste from landfill each year, reducing its footprint. But also, as you can imagine, the composition of MSW can vary considerably depending on climate, season, culture, income level, etc., reflecting different global patterns of consumption, which means that as a biorefinery feedstock, it would rarely be compositionally the same twice. So to get an idea of just how compositionally different it can be, we recreated the proportions of the different biogenic fractions of MSW found in high and low income countries. And these fractions included food, plastic, paper and cardboard, textiles, and garden waste, which are found in different proportions depending on a region's income. So, for example, food waste is a major component of MSW in low-income countries, while paper and plastic tends to dominate in high-income regions, which again is a reflection of the broader patterns of consumption and waste generation across the globe. And capturing this variability is critical for understanding how robust and flexible our microbial system needs to be. So we recreated these different fractions using representative constituents, which you can see in panel B here. So for example, for food waste, we used a mixture of vegetables, fruit, grain, and oil, whereas for plastic waste, we used a mixture of PET and PLA, PLA being polylactic acid. And then we subjected these high and low income waste compositions to an enzyme-based hydrolysis using a tailored enzyme cocktail that we knew would break down each constituent. Then post-hydrolysis, we identified and quantified the 9 most abundant monomers released from both mock wastes. And these monomers included sugars, aromatics, acid styles, and polyols, which you can see in panel C. The relative proportions aligned quite well with the initial waste compositions, which demonstrated that our tailored enzyme approach effectively captured the key components of these waste streams. But in the interest of transparency, I will caveat this work by saying that these mock compositions are very much a simplification of what you would find in real waste. And in a true industrial process, a more effective pretreatment would likely include a chemical hydrolysis step to break down any non-enzymatically liable constituents. The combination of biological and chemical pretreatment methods is a common strategy to ensure maximum substrate recovery. But for this proof of concept, we were interested in pursuing an entirely bio-based process, and this decision was made to reflect a broader interest in biotechnology research to minimize reliance on harsh chemicals, even if they are currently more effective. Okay, so of the top 9 monomers released from the mock waste compositions, Putida is natively capable of assimilating most of them, with this diagram here showing how each of these 9 substrates enters this microbe central metabolism. Those that Putida cannot natively assimilate are the sugars xylose, the aromatic terephthalic acid, or TPA, and ethylene glycol. So to address this, we engineered a strain of Putida with the necessary additional catabolic steps required for utilization of these substrates. And then we validated its ability to grow on each as a sole carbon source, which you can see here. In the case of xylose and ethylene glycol, we found that constitutively expressing these heterologous pathways was preferred for metabolic uptake. While Putida had improved growth dynamics on TPA when this pathway was regulated by a genetic feedback loop, which is turned on by the presence of protocatachoic acid, which is an intermediate in the metabolism of TPA. And this is a scheme that you saw earlier from Neil. So now that we had a strain that was engineered to be able to assimilate all of the 9 dominant waste-derived monomers, we wanted to get a better understanding of the carbon use efficiency for each of them. And by carbon use efficiency, I mean the ratio of carbon in a given substrate that partitions into biomass accumulation as opposed to CO2 efflux. And so to calculate carbon use efficiency, we coupled experimentally derived sole carbon growth data for each substrate with flux balance analysis. And the results are shown in panel A here. What we found was that carbon use efficiency varied significantly between substrates, which really highlights the importance of substrate-specific factors in determining overall process performance. But the general trend from this data was that those substrates that are more reduced, or rather have a lower nominal oxidation state of carbon, like the fatty acids, glycerol, and lactic acid, had higher efficiency ratios and thus promoted greater biomass accumulation. And the underlying metabolic explanation for these results could be that it's a virtue of the chemical energy content per unit mole carbon of a given substrate, with the more reduced ones providing more reducing power for biosynthetic processes. Or it could be related to the number of CO2 generating reactions associated with the individual catabolic assimilation pathways. The factors that play into these ratios are complex and not entirely well understood. But ultimately, for a bioprocess, this information is impactful because the synthesis of most bioproducts is tied to biomass accumulation. And the knowledge that there are inherently better and inherently worse substrates for biomass buildup and production means that we really need to approach the microbial valorization of waste streams in a strategic way, where we take into consideration the limitations of a feedstock with regards to its potential variation extremes and what that impact will be on the process economics of a potential end product. Okay, so then from here, what we next wanted to look at was the ability of our engineered Putida strain to simultaneously utilize the 9 waste-derived monomers by performing a flask growth assay in a minimal media containing equimolar concentrations of each substrate, which you can see in panel B here. And using flux balance analysis again, we then looked at how the metabolic state of the cell changes during the early, mid, and late exponential phase of cultivation. And what these snapshots show is a metabolic shift from a predominantly gluconeogenic configuration to a glycolytic one over time as the strain co-utilizes the available substrates at different rates. This strain did not demonstrate significant dioxide growth, which can likely be attributed to the reverse carbon catabolite repression mechanism used by Putida, which operates at the mRNA level instead of the protein level. And so it allows for more sensitive response to substrate availability. And overall, the takeaway message from this metabolic analysis is that when grown in the presence of complex mixed substrates, Putida can quickly and dynamically change the configuration of its central and peripheral metabolic pathways in response to changing carbon and energy availability, which in the context of a heterogeneous waste feed with changing substrate ratios and concentrations is especially important. So to further probe just how dynamic this metabolism is, we used a design of experiments approach to construct a set of 25 feed compositions using our 9 monomers to explore how our engineered strain performs across the full combinatorial space of substrate loadings that might be found in process-relevant conditions. And what we found from this work was a correlation between the amount of carbon in a given composition that can contribute to biomass and the final biomass obtained, which means that regardless of the constituents and proportions of a given composition, the strain was able to efficiently and consistently convert all its available carbon to biomass, which is particularly a particularly powerful feature for a bioprocess. So then from here, in a final experiment, what we wanted to do was look at how different feedstock compositions can impact bioproduction titer by Putida. And so for this, we focused on producing 2 very different products, PHA and interferon, which I mentioned at the beginning of the presentation. Neil also touched on interferon. So PHAs are biopolymers that represent an alternative to conventional plastics. They're renewable and biodegradable, which makes them an attractive replacement option. Whereas interferon is a high-value biotherapeutic protein used to treat a number of different diseases. And like Neil said, before we settled on using interferon, we did screen a number of other therapeutic proteins, including insulin analogs and synthetic nanobody. I will also reiterate Neil said, in that by no means do we expect that in the future people are going to be making biotherapeutics from MSW. This was just meant for the purposes of demonstrating that you can make quite disparate products in a microbial biorefinery. Okay, so we then performed bioproduction time courses for both PHA and interferon using our high income and low income hydrolysate compositions that we previously harvested from our mixed waste hydrolysis that I showed at the beginning of the presentation. The graphs in the top row show interferon production and substrate consumption over time, and the bottom row shows the same for PHA, while the graphs in the right column show the high income hydrolysate composition, the left and then the right show the low income composition. In the case of all bioproductions, Putida funneled approximately half of the available carbon from the high and low income feedstocks to biomass and product formation. So again, we're seeing a consistent relegation of carbon regardless of feed composition. But despite this, the low income condition did produce more product overall. For both products, we made approximately 5.5 mg per liter of interferon and 27% cell dry weight of PHAs. And the likely reason for this is because this composition is richer in those reduced substrates that contribute more to biomass and production. So then in a final experiment, a final step, what we wanted to do was to assess the environmental and economic impact of our proposed MSW valorization approach. And so to do this, we conducted a life cycle assessment, or LCA, and a life cycle costing, or LCC. And these tools allow us to compare our proposed biorefinery platform for PHA production against incumbent municipal waste processing strategies, including landfill, open burning, and incineration with energy recovery. And so LCA is a systematic framework used to evaluate the environmental impact of a product or process throughout its entire life cycle, from raw material acquisition through to end-of-life scenarios, whereas in contrast, LCC complements LCA by focusing on economic factors, including capital expenditures, operational costs, and potential revenues. So together, these tools are essential for understanding not only the sustainability, but also the economic viability of scaling up this microbial valorization process. So from the LCA, what we found was that the proposed Putida-based valorization of MSW bioprocess shows significant potential for reducing the carbon footprint of MSW treatment. It had about a 40 to 60% reduction in greenhouse gas emission compared to conventional methods, which is shown on the bottom axis of this graph. The LCC showed about 25 to 40% lower process cost per ton raw material compared to landfilling, which offers a strong economic incentive for adoption of this process. But it also showed a 10 to 30% higher cost compared to incineration with energy generation. And a key insight from both of these analyses was that the identification of the enzymatic hydrolysis step as the main hotspot. It contributed about 90% of the total climate change impact and 70% of the costs. And this highlights the importance of optimizing or replacing this step to enhance overall performance. So to achieve improvements in this particular step, one could explore innovative enzyme formulations, integrating different chemical pretreatment options, or leveraging high throughput process optimization. But ultimately, what these results indicate is that the use of MSW as a feedstock in a microbial-based biorefinery is a promising alternative to the current disposal options out there. And so with that, I'm just going to acknowledge everyone who worked on this particular project. So Guada, Pia, Doc, Carl, and Rosa. Also everyone in the wider Dixon group. And then thank you to IBIC and everyone for listening. And then that's it.
Brilliant. Thank you very much both for some really insightful talks there and some great examples of how we can turn that waste into wealth and really make something useful from our municipal waste and other waste streams. So I don't know if anyone's got any questions that they want to ask. We've got a few minutes. Yep, 1:45. Anything anyone wants to ask in the audience, you can either pop your hand up or the proverbial electronic hand, or put your video on, or pop a question into the chat. I see Jeremy there. Jeremy?
Yeah, hi everybody. Great talks. Thanks Neil and Michaela. Question for Neil about the post processing of PET hydrolysate. So you talked about converting MEG and terephthalic acid into other highly added value products. Have you considered taking the MEG out, because it has value in its own right, and in the future will be increasingly bioproduced in any case? So just wondered what your thoughts are about that.
Which bit? Sorry, is a bit of disruption on the line.
So I'm sorry. I'm also in a very noisy background place, so that might help. So you were looking to co-convert monoethylene glycol to and terephthalic acid. I just wondered whether you considered the option of taking the ethylene glycol out separately, given that it's a highly added value bulk
we
chemical in its own
haven't.
right.
We haven't. You could think of ways of doing that. Not all organisms can assimilate it. So the organism that we worked on, the Pseudomonas putida, consumes it natively, but doesn't convert that into energy. So it doesn't convert that into biomass. So if you leave the microbe on its own, it will break down a bit, but won't actually grow. So we've engineered it to convert it into growth and energy productions. But by the same route, you could think about knocking that out. I guess as a toxicity issue in terms of if you've got it in your milieu with your microbe as well at some sort of threshold. But that was a good point. I didn't really know the value of it, if I'm really honest, at the top of my head.
Well, it's a precursor to many other chemical products, and increasingly, I think in the future it will be bio-based in its own right. So there's an advantage in reusing it in terms of the circularity of a bio-derived material. Just something to think about, anyway.
Yep.
Perfect. Thanks for that,
Thanks.
Jeremy.
Thank you, Jeremy.
Any other questions? While we're waiting, I'll ask one. So, Mika, you were showing that you're looking at your carbon that comes out of your bioreactor, and you're looking at sort of the carbon that comes out through your monomers that you're finding, and also CO2. So I assume that's what the 100% maps onto. It's like the total amount of carbon that you've measured coming out. How does that relate to what sort of the proportion of carbon that you're able to transfer into something useful versus the carbon that's going into the feed stream? So I guess it's a bit of sort of the efficiency type question.
Yeah, so in terms of like the amount of carbon going to biomass versus product formation,
Yeah.
is that what you mean?
Yeah.
Off the top of, that's a good question. I don't think we actually, do you know, Neil, if we actually did that calculation? Because I can't think of it off the top of my head, what the ratio was there.
No, I don't think we did. I mean, I guess we're making, in the PHA scenario, we're making 30% of dry cell mass is the biopolymer. So forgetting about molecular weights, maybe, you could say that 30% of all that mass is going into your product, and the other 70% is going into the microbial mass.
Okay, so of the 50% that goes into biomass and product, 30% of it approximately is product.
Okay. And I guess anything that you convert is better than the current state of the art, where it's just going to landfill. So I think that's positive. And in terms of, just thinking about, future looking, what do you think are, I guess, two questions. One, what do you think is going to induce a step change into some of this being adopted, in terms of what are the current bottlenecks in terms of the technology being adopted from a scientific perspective, and not also then a policy/regulatory perspective?
I think finding for complex waste streams a pre-treatment method that is efficiently hydrolyzing all of the carbon down to consumable monomers for bacteria is going to be a challenge, particularly. And then sifting through what those monomers are and engineering a bacterium that can utilize all of them efficiently will be two big challenges in the future.
So maybe I'll complement the technical bit with a sort of holistic bit. I think it's a real challenge in terms of the ecosystem, in terms of finding this, or sorting, collecting and processing this residual waste biomass or waste carbon, and the infrastructure that's needed to facilitate all that. I think that's a big challenge. I think the municipal is a nice story. There's probably lower hanging fruit, industrial off streams that are probably hopefully purer. So stuff coming out of food waste, out of factories, out of brewery, for example, it's probably a bit more homogenous, should we say, a bit more consistent, and requires less challenges. I think the sectors that are doing it, I think sustainable aviation fuel and biodiesel and anaerobic digestion do deal with these spread of these kind of issues in terms of collection and sorting.
Yeah, no, I think that sounds good, and I think it's good to look at all sort of different types of waste streams, because it's not going to be one solution, one carbon source, to solve sort of the sustainable angles moving forward. We've got another question. We've got Ed here. Edward.
Yeah, hi Aileen. Yeah, this one's to Neil, to either Neil or Mika. I'm Ed Ranvier, by the way. I'm from Manchester Metropolitan University. Great talks. Thank you very much for sharing your work. I really love the idea of having a situation where you can process several waste streams at once. The only comparable technology just been mentioned is anaerobic digestion, as far as I know, and in Greater Manchester especially, there was some big challenges with that on an industrial scale. And Mika mentioned about pre-treatment processes for chemical feedstocks, and the thing which came to mind was the idea that you're working with microbes, and you're going to need a sort of, I would assume, a sort of fine balance, a tolerance for these, like a very small window of pH, for example, where these can operate. So I guess my question is with regards to what is the do these microbes have tolerances which will allow sort of flexibility in a biotechnology scenario?
Okay, do you want to go, or do you want me to?
In terms of pH, I think there's probably a smaller window for most bacteria. You'd be looking between maybe 6 and 7.5. So you'd probably have to buffer your hydrolysate if it was outside of that range. Protida is quite unique in that it has a very high tolerance for a lot of common inhibitors from pretreatments. So it's able to handle furfural and HMF quite well compared to other bacterium. But if you were to work outside of the range of its tolerance, you'd probably either have to dilute your hydrolysate, or you could engineer your bacteria to have improved tolerance to these inhibitors.
I was on mute because I was coughing and then I forgot. Rookie mistake. So thank you very much for that. So I think, given the time, I'm going to just wrap things up now. So I just wanted to say thank you very much once again to Neil and Mika for excellent presentations today. And I'm sure if anyone wanted to reach out to either of them for follow-up questions, or if anyone wanted to engage with either, looking at potential relationship builder grants, or wanted to know more about potential collaborative opportunities that we could got some money through IBIC to help accelerate or help entice you, then I'm sure they'd be happy to have a chat as well. I'd like to just say thank you to everyone for joining. Our next webinar in our IBIC series is going to be 12 o'clock, 12 to 1 on the 16th of December. And that's going to be given a lecture from James Winterburn, talking about fermentation. And I assume he'll talk about the translation of his technology into holoferm as well. And he's going to be joined by his postdoc, Pavit I'm going to pronounce that. Pavit Wongstrisho, and we'll pronounce that. Neil's laughing at me. We'll learn how to pronounce that properly on the 16th, and hope to see you there, 16th of December at 12 o'clock. Thank you very much, and enjoy the rest of your day.